Importance of enzymes. First of all, the definition of enzymes. Enzymes are biological catalysts. They speed up biological reactions without being consumed in a reaction. This means that enzymes are catalysts. They are not used up in a reaction, but they only speed up a chemical reaction and they do not alter the equilibrium. Now for the discovery of enzymes. In 1833, a French chemist, Anselme Byron, discovered the first enzyme, diastase. In 1877, German physiologist Wilhelm Kuhn first used the term enzymes. Now for the properties of enzymes. Enzymes are organic catalysts, however, they have properties of inorganic catalysts. Number two, and second, they speed up chemical reactions by 10 to raise power 6 to 10 to raise power 14 times faster. Enzymes are global proteins except ribozyme, which consists of RNA. This means that enzymes are made up of large, long polypeptide chains and they are proteinaceous in nature, except the ribozyme, which is made up of RNA. Enzymes are highly sensitive to temperature and pH changes. This means a slight change in temperature and pH can alter their function and can even denature them. Enzymes are either highly specific, for example, sucrase, or slightly less specific, carbonic anhydrase. This means that specific enzyme sucrase can only work on sucrose, but carbonic anhydrase can also work on oxygen as well as carbon dioxide. Enzymes can live in both vivo and vitro. Now for the vivo, that this means that they can live in living cells. Of course, they are found in living cells, and in vitro. That means they can also live outside this living body in test tubes, etc. Some enzymes need for cofactor for their proper activity. Those enzymes cannot work proper without their cofactor. Enzymes only speed up a chemical reaction and do not affect the equilibrium. This means after the equilibrium is maintained, the enzyme will not have any effect on the chemical reaction. Without enzymes, there would have been no existence of life. This means that enzymes are so crucial that they speed up chemical reactions so much. That is why the life is present. Without enzymes, no life would be present. Now for the human enzymes in the body. Enzymes in human body. There are certain groups of enzymes in human body which I'm going to discuss. There are approximately 1300 different enzymes found in human cell. Lipases, a group of enzymes that helps in the digestion of fat in the carbohydrates. They cause the breakdown of carbohydrates like amylase and cellulase, etc. Proteases, they act upon and proteins, example pepsin, trypsin, etc. Nucleases, they are involved in the breakdown of DNA and RNA, for example, DNases, RNases. Now, the food we consume consists of all these lipases, carbohydrates, and proteins, nucleic acids. How these are digested in our digestive system? These are helped by the help of these enzymes. Now, for the enzymes in different food industries, yes, enzymes are not only found in living cells, they are also found in different food industries. For example, food processing industries, which tenders the meat and making of syrup, fish products, dairy products, alcoholic drinks like convert sugar into ethanol and making of bread, seaweed products, leather products, and removal of hair from the animal hides and textile products. And medical and pharmaceutical are most important. For example, pancreatic trypsin treats inflammation, and microbial trypsin dissolves the blood cells. And biological washing powders are very important, and they are used in washing powders because the stains on the clothes are also organic. As you can see on my screen, there are different showing you like catalyst and biotech and diagnostic and food, beverages, animal feed, pharmaceuticals, biofuels, and household items. I mean, there are a vast variety of fields where enzymes are used. Now for the five food enzyme families. Food enzyme families are cellulase, amylase, and lactase protease. They help in different digestion of different stuff in our gut and in our digestive system, like digesting sugar, digesting carbohydrates, digesting cellulase, and digesting fat, dairy products, and protein from nuts, beans, etc. Now for the enzymes used in food industries, like in dairy product, rennet, lactase, and protease, and brewing industry like beta-glucanase, amylase, etc., and making baking like glucose, oxidase, etc., and in fruit juice like pectinase, and for making the food more tastier, more digestible, like making the juice more tasty and tendering the meat to that is properly cooked, like protease and peptin. Now for the enzymes as a diagnostic regulations. Like enzymes are also used in diagnostic of different diseases. For example, glucose oxidase in glucose estimation, and glucose uric acid, and urease in urea, cholesterol oxidase in cholesterol formation, and lipase in determination of triglycerides. Now, by using these different enzymes, we can estimate that which type of disease are present in human or in an organism. Now, for the enzymes and drug targets, they are not that drugs, they are but as a medicines, like different enzymes are used in different medicines, like in cancer medicine, like malaria, protozoa, in bacteria, in gout, in treating cancer, in antibiotics like penicillin, and for treating the HIV and SARS diseases, and HIV virus and coronaviruses, and ribozymes. An enzyme and molecule capable of acting as an enzyme. They are not actually proteins. They are made up of RNA, but they act as enzyme. Now, for they're used in ribosomes, like making the aminopeptide chains and producing different required proteins of different cells, different tissues. And now for the artificial ribosomes. They are ribosomes used in synthesis in the laboratory based on dual nature of RNAs as catalase and as information polymer. Synthesis of artificial ribosomes include mutation of natural ribozymes include enzymes, ligase, ribozymes, and allosteric enzymes, etc. Now that's my presentation end, and thank you everyone for listening.